I hope you can hear me okay. My name is Lukas Volz. I'm the professor of stroke and neuro rehabilitation at the Department of Neurology at the University of Cologne in Germany. And I'm very happy that you guys invited me to talk a little bit about our work on structural reserve and motor recovery after stroke. Before I start diving into the topic, I want to give you 1 minute of background so you know who's talking to you and why this person has such a weird accent while speaking. I trained in medicine and did my PhD work in Bochum in Germany using transcranial magnetic stimulation in the rat model to investigate mechanisms of plasticity. And then after that, switched to the Max Planck Institute for Neurological Research and also the Department of Neurology in Cologne, where I started applying transcranial magnetic stimulation to stroke patients, using also functional and structural imaging to better understand what's going on in the brain while we stimulate it and while it recovers from stroke. And this is what I then took to Santa Barbara. I did a postdoc at UCSB with Mike Zaniga and Scott Grafton at the Brain Imaging Center. And I'm showing you this because a lot of the images that you'll see stem from a specific diffusion imaging protocol that we designed in Santa Barbara and then brought to Cologne to acquire it in different stroke patient populations. So to get right to it, I was asked, or you saw one of our recent publications that was published about 2 years ago in the Annals of Clinical and Translational Neurology, entitled Corticospinal Premotor Fibers Facilitate Complex Motor Control After Stroke, and asked me to present this work. As this is part of several studies that we've published using similar methodology, I will present the findings of this study, but I will embed it into 2 or 3 other studies that A, provide context, and B, also allow for a more in-depth interpretation of the findings. So let's get started. I assume, given your background, most of us are somewhat interested in recovery of motor control after stroke. As most of you know, motor symptoms are one of the most important impairments for stroke patients. When you can't eat or dress yourself, that's always going to impact on your quality of life. And so we want to understand how recovery happens and what we can do to facilitate recovery. And what do we know about recovery of motor control after stroke? First of all, we know that it's a heavily time-dependent process and that a lot of recovery happens early on in the first couple of days to weeks after a stroke. And this is a period that's often called the critical period of heightened plasticity in which recovery is really possible. The other thing that we know, and obviously there's some debate about this, but I think most of us will agree that recovery and motor recovery is not only time dependent, but it's also highly variable. Here you see an example of Van der Vliet and colleagues' work from the Netherlands that shows in a cohort of stroke patients how the Fugel-Meyer upper extremity score representing motor dysfunction changes over time. And you can see that while weeks advance, most patients do get better. Each of these lines represent a patient here, but you see that there are vast differences in recovery. While some of those patients make almost full recovery, a bunch of them are also left with quite poor outcome. And so the question is, what explains this difference in recovery and what happens in the brain to facilitate recovery? To understand that, let's perhaps start from a first principles perspective. So we need some parts of the brain to essentially guide voluntary movements. And what we do need, for example, is the primary motor cortex, which is the main part that generates motor commands. But obviously, it doesn't do that on its own. It communicates with a network of motor regions. Here I show some cortical regions, but we can also think about subcortical motor areas that are involved in motor control. Once we have those motor commands, we also need then to essentially project them to the spinal level, to then be able to facilitate activations of muscles, of peripheral muscles, to essentially generate movements. And this is where the most common type of ischemic stroke comes in, namely subcortical strokes of around the internal capsule, middle cerebral artery infarctions that usually disrupt these descending pathways. And this is kind of a little bit of a conundrum if you think about it. These fibers are disrupted and they won't regenerate. Long axons, most people agree, won't grow back. So you don't have the possibility of new axons forming that go all the way from the brain to the spinal level. However, patients still recover. And we think this is in large part due to functional reorganization, or what the network can do to really change how motor commands are computed and then also transmitted to the spinal level. And this is what I want to talk about today. How can we use imaging, and today we'll talk about structural imaging mainly, to essentially assess what the motor network can do to reinstate motor control after stroke. So to look at that, let's turn to the concept of structural reserve. I already said one important kind of prerequisite that I think we have to agree upon for this reasoning that you'll see unfold over the next slides to make sense, namely that long axonal connections, both from the motor cortex to the spinal level, but also between distant motor regions, are unlikely to grow back in the adult post-stroke brain. If you agree on that, then changes in how the network computes and then projects motor commands to the spinal level have to rely on axons that are still there after the lesion. And this is how we operationalize structural reserve. So if you think about the structural reserve, it's those type of fiber connections that still remain intact after the lesion. And as a lot of you know, and I won't talk too much about this today, a lot of work has looked at the corticospinal tract and has shown consistently that the more intact the corticospinal tract, the main and most important output pathway of the human motor system is, the higher is the likelihood for recovery and the better the patients will do in terms of motor control after stroke. However, it doesn't stop with the CST. There are other structures that are important, and some studies have already shown that. For example, Schultz and colleagues from Hamburg have shown that connections between premotor areas and M1 in the ipsilesional hemisphere also may contribute to functional recovery of motor control, and also a beautiful work, earlier work has shown that interhemispheric connections between both hemispheres may contribute to motor recovery and motor control after stroke. So how can we conceptualize this? How can those connections help in recovery? And I like DiPino's definition of structural reserve here that is at this point more than 10 years old. But what it says is that structural reserve is actually the quantity of strategic neural pathways and relays that are spared by the lesion and that can reallocate previous or outsource new functions. So in other words, what are the parts of the network that you can use if a specific part has been damaged by stroke to essentially compensate for that damage? And we wanted to look at this because it's not trivial to map out those structural connections, especially between motor regions that are located in distinct parts of the cortex, for example. So what I want to talk to you about is how can we image structural connections in stroke patients between motor areas, but also for descending pathways. And to do that, we essentially have to use, or we use, diffusion imaging. Diffusion imaging is an MRI-based technique that allows us to quantify the diffusion of water molecules. And there are one or two important things to know about this. So first of all, it really depends on, when you quantify diffusion, what kind of voxel or part of the brain you're looking at. For example, if we look at the corticospinal tract, which is a very straight pathway of a lot of fibers that all point in the same direction, you will get very one-directional diffusion. And the orientation distribution function here that we can actually calculate from the scans in each voxel look very much like this. So you have a very dominant diffusion direction. However, this is not true, and it gets more complicated if we look at voxels that are not in these main specific big fiber tracts, such as the corticospinal tract. There, the orientation distribution functions can look way more complicated and have multiple directions. And why does this matter? It matters because the most common quantification of structural connectivity or diffusion is or are measures of anisotropy. There are different ones, and we don't have to talk about the specific differences, but there's one thing that I want to show you here. If you look at one-directional voxels, these are the blue curves here, you will get a distribution of values that is pretty high. If you look at voxels with 2 or more crossings, you will see that the expected value in healthy subjects of anisotropy goes down considerably. So in other words, we really have to be careful which voxels we're looking at to get reliable estimates of structural connectivity. This is a methodological point, and I won't go much more into that. If you're interested, I'm happy to talk about it later on. But what does this mean for stroke? There's also some implications, or there are some implications for stroke patients. Namely, if you look at lesions to one-directional voxels or tracts that run in these one-directional voxels, anisotropy, so how direct the diffusion is likely to go down. So this is quite simple. We would assume that a stroke may reduce anisotropy in those fiber pathways. However, if we look at what a stroke may do in more directional voxels, such as this one here, it's not as simple to predict what's going to happen. Anisotropy might go down, might go up, stay the same. So I don't want to bore you with more methodological details. We'll get to the results soon, but I just want to really make or highlight and emphasize that the way we quantify diffusion matters if we want to understand how structural integrity of specific parts of the brain contribute to motor recovery after stroke. So to approximate this well, we designed a diffusion spectrum imaging scan, which is kind of an evolution of the more classic DTI or diffusion tensor imaging scan to resolve multiple fiber directions. You see here this hole in the right hemisphere. This is one of our chronic stroke patients that is included in the data that we're going to talk about now. So this is just an example of how this scan looks if we then do tractography. To then analyze anisotropy or connectivity, we actually looked at the anisotropy in different compartments. So we always compared one direction, two directional, three directional voxels only to each other, and we didn't average over all voxels. Again, this is a bit technical, but this compartment-wise analysis step is an important step so that we can actually make sure we will talk about structure reserve for the remainder of the time. So what did we do? We acquired diffusion imaging data from chronic stroke patients. And to really define structure connectivity, this is the abbreviated workflow. What we did is we got whole brain generalized fraction anisotropy maps that just show how directed is diffusion in each given voxel. We then masked those voxels for white matter so that we only look at white matter voxels. We also masked out the lesion, because we don't want to look at what's happening in the lesion. We want to see what is the structure reserve, so the white matter voxels and pathways that are not affected by the lesions. And then we use our compartment mask that we designed to only look at one directional voxels mainly. And then in the last step, we define a tract, and we'll talk about descending tracts and corticocortical tracts in a second. We define a normative tract and then extract the voxel-wise diffusion information for these normative tracts. This was a lot of information, but I just wanted to give you a little background on how we calculate structural connectivity. And let's say this is the corticospinal tract, for example. Now we would then calculate the structure reserve here as the mean anisotropy across all voxels in this tract.
Your call cannot be complete.
Okay. The other thing we did in the studies that we assessed is motor impairment in our stroke patients. And we did assess different kinds of motor impairment because this is another thing that's important to note. I'm sure there are some clinicians here in the room, and as the clinicians know, different aspects of upper extremity motor function recover differently. If you look in the literature, you find something like a proximal distal gradient of recovery. But I think most of us see in clinical practice that essentially proximal movements often recover quicker than distal, more fine-grained complex hand movements. So we use two tests, the motricity index, which comprises very simple 1 degree of freedom movements such as shoulder abduction, adduction movements, and is rated. And we used a more complex test, namely the Action Research Arm test, which largely also focuses on more complex hand movements such as pinch or grip movements or grasp movements. And we will differentiate basal and complex motor control for the rest of this little talk. So what do we now do and find? So in a cohort of chronic stroke patients, the first thing we looked at was using our method that we came up with. Can we essentially can we show that what we already know is there when we see this? Can we replicate earlier findings? So the first thing was to look at the corticospinal tract. And yes, what we see using our method is what you would expect, namely that higher integrity in the corticospinal tract, so higher anisotropy, is related to better motor scores in chronic stroke patients. So congratulations, we essentially replicated what a lot of studies have already shown. If we look at what happens in these compartments of all voxels or 1 and 2 directional voxels, there is an interesting thing because if we look at young and old control subjects, we see that, for example, we see an aging effect in the directionality of the voxels, just comparing the control cohorts that differ in age. But we don't see essentially a stroke effect on these 2-directional voxels that we do see in the 1-directional voxels. Don't want to go into this in too much detail, but this could mean that aging and stroke may differentially affect these compartments of anisotropy voxels in the human brain. So what you should take away from this is that we show that essentially reduced anisotropy, so reduced structural integrity of the corticospinal tract, is associated with motor impairment. And I'm sure this is old news to a lot of you. But let's move beyond the CST. So what we did then was we looked at other extrapyramidal descending tracts, because if the CST is damaged again, you need to find a different way to communicate motor signals down to the spinal level. And what we did was to look at the rubrospinal and reticulospinal tract, and these were not damaged by the lesion. And we found correlations between the structural integrity of those tracts and basal motor scores. Here I show you the motricity index of the leg as one example. And with higher integrity showing stronger motor control. So what this indicates, and obviously we're not the first to show this, there's a whole literature on this, but it fits to the notion that extrapyramidal tracts may contribute, especially to the control of basal motor movements after stroke when the corticospinal tract is damaged. Let's now switch gears a little bit and talk about other CST fibers, because so far I've told you when the corticospinal tract is damaged, we got to compensate for that damage, looking for other tracts. And what Teresa Paul, a postdoc in my group, who's also here in the audience, I think, showed in this work or looked at was, and this is the paper that you invited me to present on, was that what about corticospinal tract fibers that originate not from primary motor cortex, but from premotor areas? Could those fibers potentially help to generate output from the cortex to the spinal level in order to circumvent the lesion? So in other words, instead of looking at the CST or thinking about the corticospinal tract as something that just originates from primary motor cortex, we now differentiated different subtracts. So fibers that come from primary motor cortex, M1, fibers that come from supplementary motor area, dorsal premotor cortex, and ventral premotor cortex. And we now ask the question, does the structural integrity, the structural reserve of those fibers relate to motor control, basal and complex motor control in, again, chronic stroke patients. And what we found looking at complex arm movements is that, again, you find positive significant correlations, meaning the stronger the structural reserve, the more structural integrity you have in the CST subtracts from primary motor cortex, but also premotor areas, the better the patients are at moving their hands. You see that these correlations are not perfect, but all of them are significantly positively associated with complex motor function. So from that, we can already take away that premotor CST output may shape complex motor control and hand control after stroke. This is not really surprising, given that we know that premotor areas are heavily involved in fine-grained and shapes of hand movements, initiation and control of what you do with your fingers and your hands. So this fits very well with what we expect also in healthy subjects. In the next step now, we looked at basal motor control, so again, the motricity index. And at first, across the whole cohort of patients, we only found a correlation with M1 fibers. And we were happy with that too, because, okay, like to do a very simple movement such as abduction, adduction, shoulder movements, or flexion, extension movements in the elbow, you might not really need all that much contribution of premotor areas, and M1 output might be sufficient. However, something interesting happened when we then looked at a subgroup of patients that essentially was severely affected and also in the chronic phase showed remaining motor deficits, so they didn't recover to a large degree. In those patients, we found that the structural integrity of those premotor tracts, all three, was significantly associated also with basal motor control. And this is important to think about again. So in healthy subjects, we wouldn't really expect this necessarily. And also, when we looked at across our whole cohort, we didn't see an association with these premotor fibers and basal motor control. However, in those severely affected patients, all of a sudden, the residual structural integrity of those descending pathways was associated with basal motor function. And this really kind of motivated us to look further, because what it could mean is that if the CST output of primary motor cortex is damaged, maybe the motor system could use some of those CST fibers that run from premotor areas to the spinal level and then may not be damaged by the lesion. Of course, this is not proof of it, but these results motivated this line of thinking. So what did we do next? We essentially said, okay, if it's really potentially a rerouting, so M1 fibers are damaged, and now we have to get motor commands rerouted to other premotor areas or the other hemispheres, so other places that have descending fibers, we have to look at what's going on the cortical level. So essentially, we took a group of chronic stroke patients and now here calculated using this very same approach, structural connectivity between premotor areas and M1, both within the affected ipsilesional hemisphere and the contralesional hemisphere. And this is work that was published in Annals of Neurology, and I guess they liked how these figures look, which is easy to do with diffusion imaging, and put this work on the cover about 3 years ago. So what did we find? Again, we looked for associations of motor control in the chronic phase after stroke with structural reserve. So how the fiber pathways between the shown areas are preserved. And what you see at first is that there are a lot of significant correlations for both basal and complex motor control. And I want you to note that they're all positive, meaning we always find that the higher the structural integrity of these connections, the better the motor control in the patients. That makes sense, right? But of course, so many positive correlations, what could that necessarily mean? This is more points to a more general aspect, right? If the patient has a quote unquote better, more intact brain independent of the stroke, maybe they recover better, right? So we wanted to look more specifically, and in the next step, we controlled for the degree of CST damage. And what happens then is quite interesting. Complex motor control, when you account for the degree of CST damage, all these associations go away. In other words, it may be hard for the motor system to really compensate extensive CST damage to facilitate complex motor control after stroke. However, when you look at basal motor function, even when you control statistically for the degree of CST damage, you find that a lot of significant associations remain. And more interestingly still, when we looked at the degree of recovery from the acute to the chronic stage in those patients, we found that this was true especially for substantially recovered patients who showed a strong, significant association between the structural reserve of the fiber bundle between bilateral primary motor cortex and basal motor function. So, in other words, the recovery of basal motor control seems to be associated with interhemispheric connectivity between bilateral M1. So, let me kind of sum up what we've discussed so far. I wanted to show you that the structural reserve enables the reorganization, especially of basal motor control after stroke. And what I've tried to show you was work that looked at the reticulospinal and rubospinal tracts, so extrapyramidal tracts that might step in and help to relay motor commands to the spinal level after corticospinal tract damage, and also premotor fibers that might help to facilitate basal motor control when the CST is severely damaged. The other thing that we saw was that especially this interhemispheric M1-M1 connection was strongly associated with basal motor control, especially in patients who underwent substantial recovery from the acute to the chronic phase. So the question is, what happens here? What happens with the information flow? And especially given this M1-M1 finding, we can ask a very simple question. There are essentially two ways this connection could help to facilitate motor recovery, right? We could have a rerouting of motor control, namely that motor signals that are essentially computed in ipsilateral or ipsilesional M1 are transmitted to the other side when the CST is damaged and may descend via non-crossing CST fibers, especially to proximal muscles. So essentially showing a detour that helps to circumvent the lesion. The other alternative might be that this connection is useful because contralesional M1 can modulate what ipsilesional M1 is doing and can help to modulate motor commands further. And we wanted to address this, and this is a paper that was just printed a couple of weeks ago, where we now, to address this question, combined structural connectivity estimates that I've shown you, that you've already seen, with measures of effective connectivity. So effective connectivity, which we measured with functional MRI. So we looked at the activation of the motor system while stroke patients performed very simple, repetitive, paretic hand movements in the scanner. And we wanted to ask the question, if the structural connectivity, the structural reserve is important in the motor system, how is it used? How does information flow change directed effective connectivity during the control of paretic hand movements? So what did we find? I'm not going to go into the details of dynamic causal modeling. This is the method we used here to estimate effective connectivity, but I will show you the results, and if there are any questions, I'm very happy to discuss the method at the end. So what we found is, here in controls, and this has been replicated in more than 10 studies that came out of our lab and other labs in the recent 10 years or more, when you move the right hand, the left M1 has to be activated, and you see these green arrows that show facilitatory input from essentially premotor areas of both hemispheres onto the left M1 that is controlling the right hand. At the same time, right M1 has to be inhibited. This is also something we've repetitively seen, so that you really get unilateral hand movements. What do we see in our patients? In our patients, we see a very similar picture, but there are some marked differences. One is that we see a little bit of reduced premotor M1 input, but more importantly, we see that the inhibitory connections, so what we see in healthy controls, namely that the active M1 that controls the moving hand inhibits the other side, turns into facilitation in those stroke patients. So we see a change in essentially the nature of this connection during movements of the paretic hand. So This, to our mind, is a first answer that is very interesting. Namely, this really argues that we see movement-dependent rerouting of motor commands from the ipsilesional to the contralesional hemisphere. Then, to go full circle, essentially, we wanted to see whether this rerouting that we see with functional data is something that relates to the degree of structural connectivity. So in other words, we've seen, or I've shown you in our previous study, that this M1-M1 connection, structural connectivity, was positively associated with motor recovery. So the more structural reserve you have between the hemispheres, the better you may be able to use the contralesional hemisphere. That was our assumption. However, disappointingly, to some degree, it's not as simple as that. So when we look at the correlation between the structural connectivity between bilateral primary motor cortex and the effective connectivity, so what the network actually integrates in terms of information during movements as measured by fMRI, we see that there's no association. So we were a bit puzzled by this result at first, and then thought to look beyond essentially the connections themselves. So look at whether there are patterns emerging when you look for correlations within the whole network between structural and effective connectivity. And indeed, we found two interesting things. One was that we found here a negative correlation between the rerouting across hemispheres and premotor M1 connectivity. What does that mean? If the structure within the ipsilesional hemisphere, the structure reserve between premotor areas and M1 was low, if that was low, then patients tended to show more of the rerouting to the other hemisphere. Again, if there is not a lot of reserve between premotor areas and M1 in the ipsilesional hemisphere, we see that the motor system tends to show more rerouting to the contralesional hemisphere. And conversely, if interhemispheric structural reserve is low, we saw more rerouting in these patients to premotor areas, here the ventral premotor cortex. And then looking at functional correlations or correlations with arm function, we saw that this rerouting was actually associated with bad recovery in patients suffering from chronic deficits, while the rerouting to premotor areas was associated with more successful recovery. This is a lot at once, but I want to summarize this and show you what I think why this is important. Essentially, what we showed was that after you have an ischemic stroke that damages the corticospinal tract, the motor system seems to change information flow on a cortical level. And there are two ways that we find in our data. One is that signals can be conveyed from ipsilesional M1 to premotor areas, and from there presumably can use the ipsilesional corticospinal tract and undamaged fibers there to project signals to the spinal level. We saw this in patients with high residual CST integrity and also low structural reserve to the other hemisphere. The other way is that primary motor cortex can relay or reroute motor commands to the contralesional hemisphere. And we saw this especially when structural reserve within the damaged hemisphere was low and the CST was severely damaged. Then the motor system seems to reroute signals to the contralesional side, potentially to access non-crossing fibers of the contralesional corticospinal tract. And in our task, we saw that this premotor rerouting is something that was associated with good recovery and good outcomes, whereas this interhemispheric rerouting wasn't associated with good outcome in our cohort of patients. However, there's an important limitation, namely that this rerouting, the green and the red arrow, was only measured during hand movements. And I've already shown you that there are marked differences between proximal and distal movements, and we have to investigate further whether proximal motor control may show different patterns of rerouting. So what I want to show you here essentially, or convince you of, is that rerouting of motor control depends on several things, namely the structural reserve of the motor system, the task that you try to do and you want the patients to recover in, and how much recovery has taken place. And to sum this up a little bit, how we could think about the role of the structural reserve in the motor system is kind of akin to how you would think about navigating traffic. So if you have a stroke that disrupts one of your major pathways that you need to bring motor commands from the cortex to the spinal level, akin to a big traffic jam on a major highway, you have to look for other routes. And the rerouting seems to go or use the alternative routes that are sufficiently still intact after or in these stroke patients. So they're not too affected by microangiopathy, neurodegeneration in our patients, but also not by the stroke lesion itself. And I think that in the future, it may be very helpful to essentially quantify and look at what is the level of structural reserve that we have to then use and target our interventions, both, for example, neurostimulation, but also training, to really exploit what we have in structural reserve and not try to train something where the patient's brain doesn't even have the structural reserve that's necessary to sustain essentially this type of motor control. So with that, I want to sum up. I hope I could show you and hopefully convince you that it makes sense to look at the structural reserve of specific motor tracts and that those tracts may enable task-specific motor recovery. I wanted to show you that there are descending motor pathways that we found may be especially important in facilitating basal motor control. So basal motor control seems to be more easily compensated when the CST is damaged. And I've outlined two examples, the reticulospinal, rubospinal, so extrapyramidal tracts, and premotor CST fibers. And that by combining assessments of structural and effective connectivity, we can actually paint a fuller picture of what's going on. And we can look at essentially how does information integration, so rerouting of motor commands via those alternative pathways, how does that happen in patients? How does that lead to good functional recovery? And ultimately, are there things that we can do to facilitate these processes? And with that, I'll skip our next step where we now are expanding the characterization of the motor network for the sake of time. And I want to thank the wonderful people of my research group that do most of this work that you've seen here and that it's a pleasure to work with. And I want to thank you for your attention and looking forward to your questions. Are there any questions from the audience?
Can you hear me?
Yes, I can hear you.
All right. Question number one. We see FA increase. How reliably you can say the tract restore? Because it could be a hindrance in a paradoxal. If the water diffusion got hindered and it's got nothing to do with the tract. So we see the increase in FA. It doesn't represent the tract restore.
Absolutely.
Number two, it's completely, misleading to look at the tract and assess the whole thing. That's its recovery happening. Number three, I don't know whether you can trust the bolt function signal in a patient who have vascular disease, who have a stroke, and you are measuring the bolt response based on their pathology and trusting what the bolt signal says. It may be confounding. So how do you assess the structural reserve other than bolt?
Okay. So three great, important, and very different questions. Let me start with question number one. You asked how we can interpret FA increases. We cannot at all. I don't think that an increase in FA over time shows any remodeling of the tract. That also was not what I was talking about or wanted to show here. Essentially, what has been shown consistently is that if you damage a tract, then FA will change over time within this tract, and that it will go down usually after a damage if you talk about one directional voxels. What I showed you here were not longitudinal changes in FA. It was cross-sectional measures. So patients that have higher FA in those important motor tracts do better in terms of motor control. I think this is an important point. It's great that you asked this because it's absolutely right. Changes in FA over time, longitudinally, in large tracts, are a very difficult topic because FA is something that obviously doesn't have perfect test-retest reliability, and is something that you have to interpret very carefully. Hence, our compartment-wise analysis approach, also leaving out the lesion because there, as you mentioned, also FA can change in all kinds of ways. So I want to emphasize measuring the structural reserve is getting a measure of structural integrity of motor tracts. And what we've shown and what we see is not longitudinal changes over time, but between subject differences cross-sectionally. Let me now jump to question 3. You were asking about the Volz signal in stroke patients with changes in the vasculature. And this is an important question, and there is essentially a whole literature on this, of course. Others and ourselves have used fMRI quite consistently. And yes, of course, you get different Volz signal in stroke patients, even when you do a simple task. However, at this point, there are very good studies in larger cohorts of patients and meta-analyses that show consistencies across various cohorts of stroke patients, especially if you look at simple tasks that essentially create very robust bolt activation patterns. What I'm talking about is that these block design fMRI tasks of repetitive movements elicit quite drastic changes in bolt signal, up to 5% of the signal changes. Doesn't sound like much, but in the world of fMRI, that's a lot of signal change. If you compare this to other domains, cognitive domains, where you often get less than 1% of change between baseline and your task. So in other words, yes, assessing bolt signal in stroke patients is something to also do very carefully. However, it's not that it's completely unreliable, and a lot of studies have shown this already. To clarify, we don't use the bolt signal to assess the structural reserve. This we do using diffusion imaging. We use the bolt signal to then look at the activation and effective connectivity of the motor system while you do a certain movement. We want to see the system in action. And yes, I fully appreciate your caveat there that we have to be careful to not overinterpret bolt signal in stroke patients. And please help me with the second question. What was your second of the 3 questions?
I forgot myself.
Please let me know if it comes back.
What I've encountered is, this is just a ischemic side of the stroke. What I'm encountering is a hemorrhagic stroke. So I keep asking this question: what happened when hematoma resolved?
Yeah.
What happened to those tracts that were not showing up at the first time point, but they shows up at later time point? Are those tracts functional, or they are just there, or they were never damaged to start with?
Yeah, it's a very good question, and I think both is possible and really depends on the patient. There's another thing that we often see, and I'm sure you see this clinically, when you have small lesions that are in very strategic and tight spaces, such as the internal capsulate or the brainstem. We often see this with secondary swelling, even ischemia. So we also obviously treat hemorrhagic strokes. The data that I showed you here were all ischemic strokes because, as you highlighted, the mechanisms of recovery and what happens are different, and the resolution of the hematoma and what happens then is more complicated than in ischemia. That's why to make our life a bit easier and look at the mechanistic underpinnings of recovery, we here only focus on ischemic stroke patients. Having said that, like if you have the hematoma, assessing, for example, structural connectivity in the hematoma is not going to tell you much, obviously, because diffusion is changed by all the processes that are going on there. So I think what we would have to do is to look at the tracts in a later phase when the hematoma is fully resolved and see whether longitudinally in tracts close to the hematoma there is a change in the structural integrity. I'm not aware that this has been looked at or done. I don't think so, because this would mean that you have to longitudinally scan acutely hemorrhagic stroke patients multiple time points with high resolution scans. But it's a good and interesting question.
Okay, one last question before I take anybody else's time is fiber dispersion. What do you think about fiber dispersion? Like, for example, CST, just go from top to bottom, top to down. And when you see IPSI versus contrast CST in a patient and you see fiber dispersion, is this really happening in the real life, or is just technique limitation that, diffusion is based on model, the tracks are derived by model, and if it doesn't find it, you just pick up the next voxel and continue the track. Is it really representing?
That's a good question. I wouldn't. So as you could tell probably by my long and hopefully not too boring explanation of our methods in the beginning, I share your skepticism when we look at single fibers that tractography gives you, especially if you do this in stroke in brain of stroke patients, because you not only have the stroke, most of the patients also have some kind of small vessel disease. They have some kind of atrophy, some neurodegenerative process. So these are not your typical young adult brains in which the tractography algorithms were developed, right? So if you talk about tractography in a single stroke patient data set, I would be careful to overinterpret the dispersion that you see there. That's why when we try to quantify structural connectivity, we define those tracts in normative templates. So usually we use, for example, human connectome project data. We use hundreds of diffusion data sets from healthy controls, very carefully define our tracts in this aggregate of several hundred, and then use this tract template and project it into this patient-specific data and just extract the voxels from the tract from the single patient. And as a second thing, we use this compartment-specific analysis approach that we developed at this point 8 years ago to kind of circumvent or lessen the likelihood of running into what you said, namely that if you just have the next voxel where there's chaotic stuff going on, you might measure something that's not there. So sorry for the long answer, but I think it's an important question, and you're absolutely right. We have to be careful with which voxels we look at when we quantify diffusion, and this is why our approach has turned out the way it did, because just looking at tractography in those brains can be misleading and should be done with care. I see one more question.
Thank you.
Thank you. By Naveen El Nahas. Yes, please.
Yes. Thank you, Dr. Volz, for your very interesting and great work. I just noticed through your presentation that you never mentioned that there is inhibition from the intact motor areas to the lesioned ones. Does this now refute this hypothesis altogether?
It's a very good question. You're talking about the inhibition that we see in the dynamic causal modeling, I assume. Namely, let me go to the slide. I think what you're talking about is this here, right? So I don't think that this is enough to refute the hypothesis that there are changes in inhibition. I think what is happening is dependent on how we measure it, unfortunately. We do other work where we use TMS, for example, and also not just TMS markers that we assess at rest, but for example, we probe what the motor system does during movement preparation versus movement execution using TMS. And there we see marked changes in inhibition. And usually in the chronic phase, we see that there is sustained disinhibition. So when we would expect inhibition from the lesional to the contralesional side in healthy controls, we see some types of disinhibition, so less of this inhibition in chronic stroke patients. We've observed this for different TMS paradigms and also dynamic causal modeling with fMRI consistently, and obviously other labs have too. How this contributes, these changes in inhibition in the early phase to recovery, that is a very, very interesting and more difficult question. And to be honest, I don't have a complete consistent answer to this because I think we don't really know yet. It depends on what kind of inhibition between which regions you're talking about, how severely the patient is affected, where the lesion is, and what motor function you're probing. I know this might be somewhat of a disappointing answer, but I hope it answers your question.
Yes, thank you. This makes TMS protocols more complicated even.
Yes, it does. They're strongly task dependent and time dependent. So
Yes.
it's really, it's not straightforward, but it's, on the other hand, obviously, we're talking about the human brain and one of the most fascinating capacities, right? Namely, to recover control after an irreversible damage has occurred to very important specialized structures. Namely, the reorganization, how the human brain, if you have a 60-year-old, 70-year-old, 80-year-old stroke patient and they recover function, and you know that, this goes beyond just resolution of the penumbra, that goes beyond just resolution of swelling, and there is reorganization of the networks happening. That's to my mind quite fascinating and is something that we really have to understand better in order to facilitate recovery more in the future.
Thank you.
There's one more question in the chat, which was, is the structure reserve part of the penumbra? It's an interesting way of phrasing it. I wouldn't say that. I think the structure reserve is independent of the penumbra. What the structure reserve essentially is the blueprint, essentially what you have left, which the motor system can use, the structural connections that the motor system can use to change information flow and adapt its motor control policies. So it means that and I think to some degree, it's one of the explanations why younger patients with more intact brains, otherwise independent of the stroke, might recover better compared to stroke patients that have a similar lesion but are older and have less damage otherwise, because you just have more reserve, more residual computational capacity that you can essentially use to compensate for the lesion itself, if that makes sense.
I got a question for you, Dr. Lukas. It's great to get it. This presentation's been just so amazing for us. We started this journal club for stroke recoveries not that long ago, and so we really wanted to hear from some of the cutting-edge scientists around the world in this area. So we really appreciate the opportunity to hear on your great work overall. I was wondering, so some of the other wide matter tract pathways that you speak about that are probably important for that reserve and the ability to still be able to contribute to motor function. Also, it seems to me, are involved maybe in spasticity. And
yes.
So I wondered if you might comment or think about, so are we dealing with a situation where there's both, the dual-edged sword, so to speak, of both positive and negative consequences of relying on these other pathways?
First of all, thank you for your kind words and kind feedback. I'm really happy and honored to talk to all of you. And obviously, as you know, when you work for a couple of years on topics, you're always happy to talk about them. So thanks for giving me the opportunity. I think it's an amazing question. And I think what you're getting at is some of the reasons. I didn't go into this because some of our other works looks more at functional network changes, and there we often find what we don't find for structure. Here, what we find in the structure is that there's always positive associations between structure reserve and motor outcome. We have yet to find across hundreds of correlations over the years one negative association, meaning if you have a more intact pathway, patients do worse. We haven't found a single one of those. With function, and now I'm going to get to your question, with function we have found this, namely that there are functional configurations that are associated with bad outcomes and also bad behavior in certain ways, worse motor control. And a term that you'll often see is maladaptive plasticity. So yes, I think there are changes that can occur in the network that actually lead to non-optimal behavior. And I think you're right. If we think about the motor system, especially basal motor control, spasticity is one of those instances because, as you said, we don't understand spasticity fully, but as we know, reliance on alternative tracts is associated with more spasticity. And so it may be a really fine line to look at or to see or understand what determines whether the reliance on these alternative pathways may result in increased spasticity or whether there may be a way of the system to use these pathways without creating this ubiquitous increase in muscle tone that we call spasticity that limits movements. And I think one way forward is to really differentiate more complex and basal movements. So if you look at the literature, what we often see is that people use the Fugl-Meyer score, for example, and say this is motor recovery. Or you do a composite motor score, you calculate a PCA across a bunch of tests that you do, and then you say this is motor recovery. Well, of course, this is valid in a lot of ways. I think we will gloss over questions exactly like yours. Like spasticity plays a role that is different for let's say fine-grained finger movements and shoulder abduction and adduction movements. And I think focusing really on distinct aspects of motor control will help us to get a little further. And finally, I mean, there is great work, for example, coming from Chicago that looks at EMG activity, right? So I think this is another idea or another way to get at this is I've shown you multimodal imaging today. We haven't talked about TMS, but I think looking at EMG signals is very interesting to understand whether some of those changes are really helpful or whether they may induce spasticity. And I think you're absolutely right. That is a possibility and one way that maladaptation can happen.
Because I think about how, it's obvious you be thinking about interventions like TMS or others to see if you could take advantage of the structural reserve that does exist, activate more pathways, try to increase electrical signaling down the more clear pathways to get around the blockages.
Yes, exactly.
And what happens, obviously that'll be very interesting if you're doing that kind of work, and we look forward to it. But I also wonder about the adverse consequences too.
Yeah. You're absolutely right. And I think just to comment on this, we are doing this work. And so we've shown in a proof of principle study that facilitating ipsilesional M1 helps across subjects, but there is a lot of inter-individual variability. And I think the inter-individual variability to brain stimulation protocols is to a large degree responsible for the fact that the clinical trials we've seen, I mean, they're limited. We don't have endless clinical trials on non-invasive brain stimulation to enhance stroke recovery, but the ones that we've seen have been negative. And the negative trials, I think, if you look at what's happening, inter-individual susceptibility to the interventions is one of the big issues. And I think there are two ways forward. One is one that's already happening, that is looking at what is the functional architecture of the system and stimulating in an activity-dependent way. So only mess with the system at a certain point when you know what it's doing at that moment. And the second one, you really picked up on what I'm getting at here with all this work, is to not only look at the functional state, because the functional state is noisy, which goes back to the bold question, but also to look at the structure reserve, because that's less noisy and it's something that we can more reliably quantify. And if we want to modulate the networks, let's try to do it in a way where we use what's left.
Fascinating. Thank you.
One last question. Would you recommend running a multi-shell or a single shell DTI?
Very good question. I would definitely recommend running a multi-shell scan. Our DSI scans are multi-shell data, and this is important because, especially in those old brains that we usually look at, I'm sorry. I hope this is not offending anyone, but like obviously the majority of our stroke patients are not young, healthy adults, and so diffusion becomes even more complex. So if you have the resources and the time to scan multi-shell acquisition protocols, please do. We are big diffusion spectrum imaging, so DSI fans. I don't think that's the only thing that exists. There are other good ways to scan diffusion, and there's a lot happening. But yes, please scan multiple B values, so multi-shell diffusion. You'll thank yourself later if you build a database with that.
Thank you.
We're also pleased to see that you could measure cerebrospinal tract pathways. It's not been easy,
No.
to do so. So it's great to see more. We might love to reach out to you to see to get your help as we've had.
Please do. It's really, I mean, so the protocol that we use to assess this is something that we built back in the day in Santa Barbara when I was with Scott Grafton there at UCSB at the Brain Imaging Center, and the goal was to bring this down so that it's feasible in clinical populations. Our scan takes 11 minutes, and we are now trying to advance this. This is a scan we've now used in, I don't know, 5, 6, 7 publications that are out there that you can see. And we are, with compressed sensing, we can bring the scanning time down, and we will do so in the future. So I think it's really there's a lot happening at the moment, and I think it's really worthwhile to think about how you want to assess that. And I'm happy to chat more offline if that helps.
Thank you. Thank you so much. Anybody else with a question? Must be late there, so we thank you for
Yeah, thank you, and thanks for your attention and time. And perhaps we talk again soon. And I wish you a great day over there in Texas.
Thank you so much. Thank you.
Thank you.
Take
Bye.
care.
the so-called obesity, adiposity, fat addiction, they're all transitions. It is possible that in this condition, a metabolic anomaly exists caused by a lack of that enzyme, which liberates fat from fat cells and returns it to the general circulation. This deficiency moved out of risk. So keep in mind, by the way, that they knew virtually nothing about endocrinology in 1908, about receptors, about hormones. You couldn't measure hormone levels in the blood until the 1960s. They're guessing based just on what they know about fat tissue, about the use of fat, and what they see in their patients. So in 1909, 1910, we get back to Gustav von Bergman. As I told you, he had rejected the energy balance paradigm as being meaningful, even though he did think that there were cases of people who got fat by overeating, because he knew that you could overfeed cattle, livestock, and force them to get fat. So he would not reject that idea. But he said two other theories have to be taken into account. Okay, theories explaining fat deposition from disorders of intermediary metabolism. As in diabetes, one thinks either of an inability to burn sugar or of an increased formation of sugar. So too in obesity is conceivable an inability to destroy fat, which would necessarily lead to fat deposition, or else an easier conversion of other nutrients into fat. And then the second, another series sees this primary element, as it were, in the proliferating fat tissue itself, lipomatous tendency. We know the active increase of fat tissues in lipoma also is universal lipomatosis. Such a tendency towards proliferation of the fat tissue can be determined in the germ and the genes as a hereditary disposition, no different than growth forms of the skeleton, than inherited dispositions of other organs. It can be added as a pathological growth form, no differently than an acromegaly, which is gigantism, than multiple lipomas, etc. The cells fetch the fat, and what causal nexus remains admittedly unknown wherever they can get it. Now we have a hypothesis where the fat cells are at the center of the universe. You want to know why people accumulate excess fat, you look at the fat tissue, because it might be taking what it needs to grow, and then you have to ask what regulates its growth. Okay, and then over the coming decade, various observations are made that support this hypothesis. They cannot be explained by energy balance. One of them is the existence of a rare lipodystrophy known as progressive lipodystrophy. Mostly women, in these cases, the women become emaciated above the waist. It's called progressive because the emaciation often moves downward, beginning at their forehead. And then it stops at the waist, and below the waist, they become obese. So you have what looks like energy imbalance above the waist and below the waist, but they're going in opposite directions. In one case, there's no fat tissue. In the other case, below the waist, there's excess fat tissue. You could explain that as a fat storage disorder that's connected somehow with the central nervous system. You cannot explain it by energy balance. And then this case, which resonated in the literature for 20 years, two cases actually, both published in 2015 in the same journal. A young girl at age 12 gets a abscess on the back of the hand, and that abscess is removed, and the surgeons cover the skin on the back of the hand, a graft taken from her abdomen. Okay, so this is reported by James Strandberg, a Swedish dermatologist writing in the German literature. In a 12-year-old girl, to cover a defect on the back of the hand, a pedicle transplantation, skin graft was performed. The tissue was taken from the abdominal skin. Initially, the cosmetic result was good, but as the patient grew older and simultaneously gained weight, the transplant grew and showed the appearance depicted in the figure when the patient was 35 years old. These may be two different patients, but you can see the effect of the fat tissue taken from the abdomen. And the message was, what is it about abdominal fat tissue that causes fat storage in a person who is getting fatter? That is not the case with the other skin on the hand. By 1920, so there's a pause for the First World War. After the First World War, the world is dealing with the flu epidemic. The Germans and Austrians are dealing with the devastation that followed losing a world war. In 1920, Carl von Noorden, who created the energy balance hypothesis, writes a textbook on dietetics, and in this he discusses with great knowledge von Bergmann's theory. And he says, undoubtedly, von Bergmann's theory promises many new insights. However, it's still not sufficiently established to shake the previously valid doctrine, that's his doctrine, which emphasized the behavior of the energy balance and its influence by the thyroid gland. For the time being, we would do well to adhere to the old doctrine. Okay, this is precisely the wrong thing to say. In a field of science, if you have two hypotheses that are equally valid or invalid, you treat them equally. You don't adhere to one while you patiently wait for the other one to build, okay? Von Norden, with this statement, sets the science of obesity back arguably 20 years. In the 1920s, science of endocrinology begins to emerge. So the energy balance hypothesis was proposed because there were no other alternatives. Okay, they knew about thyroid hormones. They knew a little bit about sex hormones. In 1921, insulin was discovered, then glucagon is discovered, then growth hormone is discovered. The term hormones is only created, I forget if it was 1905 or 1912, by the British endocrinologists. So as this is evolving, the endocrinologists are coming on board with obesity being an endocrine or hormonal disorder. Arthur Beadle was the father of modern endocrinology. He wrote the very first textbook. It's published in 1907. In 1922, he said all endocrine glands that influence metabolism regulate the amount of fat deposition and dictate fat distribution. Okay, the influence of insulin on fat storage is being understood very slowly. Remember, insulin was only discovered in 1921. Wilhelm Falter, who wrote the second textbook on endocrinology, and Falter was a student of von Norden's. Falter in 1916 or 17 said basically that the fat in one needs a functionally capable islet organ, a functioning pancreas. Because in type 1 diabetes, without a functioning pancreas, you cannot store excess fat. And then he tests this hypothesis by using insulin to put fat on chronically emaciated patients. In this case, all women. You give them insulin therapy. Yes, they get hungrier, but not because their blood sugar lowers and their fat storage goes up. By the 1926 to 31, research led by Ernst Wertheimer, then a German biochemist, physiologist, later an Israeli physiologist, published a series of 10 papers. It has been found that the mobilization of fat depots and the resulting phenomena, particularly the development of fatty liver, primarily depend on the central nervous system. The CNS is directly regulating fat storage in what's called peripheral metabolism. It is now an established fact that the endocrine system also plays a role in the regulation, just as it does in the case of the glycogen sugar metabolism. In 1927, the first genetic model of obesity is created at Stanford University. The expenses, Charles Danforth, the experiments show clearly that with these animals, it's neither the environment nor the abundance of food that primarily determines the production of fat. On an ordinary diet, mice with the AY gene lay down fat to an extent greatly in excess of control animals on the same diet. The textbook science shifts. What you learn studying the history of medicine is that the person who writes the textbooks determines what people believe. So in 1900 to 1910, von Norden wrote the Nature textbook chapters. By the late 1920s, von Bergmann is writing the main textbook chapters, and now he's basically disseminating what he thinks of as the correct hypothesis, but it is becoming conventional wisdom. The primary focus is not an overall balance, but on tissue behavior, which is itself influenced by central nervous regulation on one side and endocrine regulation on the other. This emphasis, neither more nor less, is what the hypothesis of the liberal homodyst tendency aims to highlight. However, obesity remains fundamentally an issue of intermediate metabolism. That's what your body does with what you eat, not how much you eat or how much you expend. And then he says, such observations reveal the disconnect between simplistic moral judgments about overeating and laziness and the often contradictory patient histories. It seems just as illogical to say, child, you shoot up in height because you eat too much or you exercise too little, or you have remained small because you play too much sport. It's making the analogy with any other growth phenomenon. The body always finds what it needs to grow or store fat. In individuals with a tendency towards obesity, the body even conserves fat within the annual energy balance, similar to how it supports growth tendencies without the precise measurements for metabolic research or detecting. When a child is growing taller, he's in positive energy balance. Okay, his body mass is increasing, but you wouldn't blame it on eating too much, you would blame it on growth. The Lipomatous tendency lipophilia takes central stage. Julius Bauer, by the 1920s, an Austrian geneticist and endocrinologist, arguably the leading Austrian expert on genetics, endocrinology, and chronic disease. He's written a book that the New England Journal of Medicine has called a monumental work. By 1929, he's giving the Society for Digestive and Metabolic Diseases, which is the leading gastroenterology society in Germany, holds its annual conference. They have one talk on obesity given by Julius Bauer. They have one talk on emaciation or leanness given by Siegfried Tannhauser. All the leading authorities are there. And Bauer argues basically for what must have been 2 hours that obesity is a neuroendocrine constitutional disorder, and that to discuss it as an energy balance disorder tells you nothing. Never can even a proven disturbance of the energetic balance in favor of energy intake explain why the fat comes to be deposited precisely at certain body regions. There must be local differences of the skin with regard to its disposition to fat deposition. There must therefore exist differences of the lipomatous or lipogenic tendency of the lipophilia, which is a term he embraces, which alongside the general metabolic conditions prevailing in the organism co-determine the local deposition of fat. One should really think about this establishment as so evident, so self-evident that it would not even require special emphasis. He says, like a malignant tumor or like the fetus, the uterus or the breasts of a pregnant woman, the abnormal lipophilic tissue seizes on foodstuffs, even in the case of undernutrition. It maintains its stock and may increase it independent of the requirements of the organism. A sort of anarchy exists. The adipose tissue lives for itself and does not fit into the precisely regulated management of the whole organism. Under hormonal influences, it's under nervous system regulation. I'll skip over this. By 1930, remember, growth hormone was discovered, I think it was 1921. By the late 1920s, American researchers under Harvey Cushing, for whom Cushing syndrome was named, are studying the effect of growth hormones in young animals. They take a pair of English bulldogs from puppydom, and they inject one of the dogs with growth hormone, and it gets larger. You can see the skeletons. And it says the experimental animal grew rapidly and developed marked aqua enlargement. Many of the symptoms and signs of clinical acromegaly became apparent, including a voracious appetite. Nobody would consider growth hormone an appetite hormone, but it drives growth, and growth drives appetite. That was the point. Okay, the role of the fat tissue in intermediate metabolism is worked out in the mid-1930s, beginning in 1935 by Rudolf Schonheimer, another German Jewish emigre. He comes to New York. He gets a job at Harold Urey's laboratory. Urey has just discovered deuterium, for which he will win the Nobel Prize. Schonheimer and Rittenberg figure out how to use deuterium as a tracer to study intermediate metabolism, so where food goes after you eat it. And in their very first paper on fat metabolism, they report that the experiments indicated that the largest part of the dietary fat, even when it is present in small quantities, is deposited in the fat tissues before it is utilized. So you eat dietary fat and then you store it. And then you let it out of storage to burn. And this was known by 1935, and I'm going to come back to it. I'm talking about German and Austrian medical science being at the height of basically the best medical science in the world, but it's worth knowing that these ideas were not localized to Germany and Austria. Okay, the leading Swiss Swiss physician and nutritional physiologist Alfred Giegel in 1928 already had said all obesity is essentially endogenous and based on the disease of an organ or organ system. The calorie theory can only play a minor role in metabolic issues. In the UK, you could find discussions at the Royal Society of Medicine in which the endocrinologists are arguing that obesity is clearly an endocrine disorder. This is Harold Gardner Hill, that the storage powers of the body for food, salt, and water depend on endocrine influences appear most likely. And it seems to me that it's an endocrine makeup of the individual, either inherited or acquired, especially in the degree of activity of the anabolic or storage group of glands, that the solution of our problem in obesity lies. There's no evidence that they were reading the German literature by the 1930s, but the argument is being made nonetheless. The leading Hungarian internist, Giza Hetenyi, in 1936 publishes an article in the German literature, basically arguing for an neuroendocrine hypothesis of obesity, for von Bergmann's argument. This hypothesis in his experiments, he says, lists the question of the origin of obesity out of the dead end of the energetic view, i.e. what we have believed, grew up believing, and lead it into an area where through further work, the final solution of the problem may yet be hoped for. By the same year, the leading Soviet physiologist, Samuel Leitz, pretty much says the same thing. I won't read it because we're running out of time, but in the progressive obesity stage, an increase in liponeogenesis from carbohydrates, delay in fat mobilization from its depot. The former can be caused by hyperfunction of the islet apparatus, the pancreas, the latter by changes in reactivity of the vegetative sensors in the peripheral nervous system. And then it spreads to the United States. Russell Wilder was the leading obesity diabetes researcher at the Mayo Clinic. In 1932, he had written about obesity and paid no attention to this. But by 1938, he's writing a column in a journal on diseases and metabolism and nutrition reviews of certain recent contributions. He has to read all the literature. So he reads Hetenyi's article and discusses the theory. Hetenyi recently advanced the following evidence in support of von Bergmann's theory, according to which the tissues of obese persons possess an increased lipophilia. Seems to me that this conception deserves attentive consideration. The effect after meals withdrawing from the circulation even a little more fat than usual might well account both for the delayed sense of satiety and for the frequently abnormal taste for carbohydrate encountered in obese persons. 1940, Hugo Roni, who's a Hungarian emigre working at Northwestern University, ran the endocrinology clinic there, writes the first monograph, English language monograph on obesity. Only thing published previously was translations of von Noorden's work. And in Roni's monograph, he says the lipophilia theory of obesity, according to which a primarily increased affinity of all fat tissues to fat would be the essential cause of obesity, is now more or less fully accepted, chiefly in Germany by a number of leading investigators, they name some. And then in 1941, Julius Bauer, who has had to flee Austria in 1938, he's a Jewish researcher. He had run afoul of the Nazis 4 years earlier when he criticized their use of genetics to defend their social policies. Bauer is now living in Hollywood, California, hence the red identification. And he writes a review in the Archives of Internal Medicine that is a critique of the current energy theory of obesity. Says the usual conception of obesity, particularly in the US, is that it's always caused by an imbalance between intake and expenditure. This conception has been substantiated chiefly by Newberg and Johnston, who I'll get to, and almost universally accepted on account of its promulgation by leading authorities, yet is not satisfactory for the following reasons, and then he lists them. It's got 9 or 10 reasons, and then he discusses the lipophilia hypothesis. There's a 20-page review article, arguably the longest, densest, most thoughtful discussion of obesity ever published in the English language literature. In 1948, we're already ahead of the time, Ernst Wertheimer writes the seminal article on fat metabolism. Okay, Ernst Wertheimer is now considered the father of the field of fat metabolism. It's called The Physiology of Adipose Tissue. It's published in Physiology Reviews. He says the classical theory that fat is deposited in the adipose tissue only when given in excess of the caloric requirement has been finally disproved by the experiments of Schonheimer and Rittenberg. mobilization and deposition of fat go on continuously without regard to the nutritional state of the animal. Okay, think about it. If people can get fat even without eating a lot, then they can get fat independent of their nutritional state. The balance between deposition and mobilization must thus be controlled by a factor acting directly on the fats. Okay? And then this vanishes. That's the history of the adiposity. And then we shift back to what we grew up believing. Okay? And we shift back primarily because of one researcher. What's fascinating reading the literature is how much individual researchers could control what was believed. Okay, so up until 1915, if you wanted to understand medical science, you had to read the German literature. After World War I, the American medical establishment, scientific establishment, started arguing for the production of journals and English language literature that could compete because they thought we were going to go back to the war against the Germans, and we don't have to rely on German science when we do it. So after World War I, American doctors stopped training in Germany, and they pretty much stopped reading the literature. And by the 1930s, the American literature is beginning to discuss obesity, but at a level in which it was discussed in Germany and Austria in the 1900s. Okay, so we are 30 years behind, and one man, Lewis Harry Newberg of the University of Michigan, comes along and publishes 3 papers based on the same data with 7 subjects, saying that he has proven experimentally that obesity is not endogenous, it's not constitutional, and that it is always caused by eating too much. The evidence is like this. Basically, he takes one lean subject and what looks like half a dozen, it's hard to tell, subjects, and he measures their energy expenditure, estimates it, then he feeds them considerably less energy than they're eating, and they lose weight, which isn't surprising. The laws of thermodynamics demand it. And he says, therefore, obesity is not constitutional. Endogenous obesity, which has become a synonym for constitutional obesity, is a misconception. It says, in conclusion, we wish to commit ourselves to the statement that obesity is never directly caused by abnormal metabolism, but that it's always due to food habits not adjusted to the metabolic requirement, i.e. eating too much, the ingestion of more food than is normally needed, or the failure to reduce your intake in response to a lower requirement. He's got to explain. If you're going to blame obesity on eating too much, you have to explain why people who eat too much don't rein in their appetites. Especially, remember, I said if you're gaining 20 excess calories of fat a day, for storing 20 excess calories of fat a day, that's going to be 20 pounds in a decade. So it's not a lot that you have to rein in to prevent this positive energy balance from happening. So the question is, how do you explain that? Okay, it's caused by eating too much, but why don't people who suffer, who live with this burden of obesity, not eat too much? And Newberg says obese persons may divide into 2 groups. The first group is by far the larger, and then the laying on of fat is the outcome of a habit through long training. They, or we, have come to require stimuli of greater intensity before they feel satisfied, or else they deliberately disregard the warnings in order to continue a little longer the pleasures that come with eating. In other words, a combination of weak will and a pleasure-seeking outlook upon life lays the background for the condition. The mental makeup of these people resembles that of chronic alcoholics. Okay, Newberg single-handedly takes obesity, this complex science which is leaning towards obesity as a constitutional neuroendocrine disorder, and turns it into a psychological behavioral defect. This is where fat shaming is born, almost literally. Prior to this, I mean, there are always doctors who are willing to make fun of their fat patients. Newberg basically institutionalized. Okay, in 1942, after Bauer writes the most comprehensive, critical, thoughtful review of obesity in English language literature, Newberg replies with a 60-page article in the same journal, in which he says the suggestion von Bergman, he spells the name incorrectly, so volubly defended by Bauer that the fat cells of obese persons possess an abnormally great avidity for fat and exaggerated capacity for retaining it, finds no support in experiments designed to test its validity. He discusses none of the observations I've discussed. Bodybuilders inherited, he said, obesity is not. He says what's passed on from parents to children is the habits of the table, basically overeating. Summary, no internal secretion is capable of so changing the metabolism that the total amount of fat in the body will increase unless the inflow of calories is greater than the outflow. And then he has to talk about the implications again. Why would people continue to overeat when the burden of obesity is the reward for doing it? And he said, well, no attempt is made to discuss the ultimate origins of behavior. The understanding of obesity will be advanced by making a list of proximate causes. Overemphasis by a parent of the importance of food, gratification obtained from the flavors of food, the feeling of repose and comfort produced by a full stomach, temporary respite from anguish caused by intellectual, social, or sexual failure. The food habits of youth, disabling disease with its lessened energy requirement. This sounds ludicrous, and yet it is fundamentally the same thinking at the basis now of how we discuss ultra-processed foods. What do? They cause us to eat too much. They cause, in effect, food addiction. We can't say no. We can't stop. They give us too much pleasure. The exact same thinking is an exact result of thinking of obesity as an energy balance disorder. These are the references, the citations for the two papers. Bauer is in green at the bottom. His 1941 paper was published 10 times by 1959 and then never again. Newberg's 1942 response was a 7th cited paper on obesity published in 1940s. It was the first one that was discussing the pathogenesis of obesity. All the other were methodology papers. And then in 1944, Newberg follows up with a second paper on energy metabolism. And that one is the 9th most cited paper of that decade. This is the death of the fat storage disorder hypothesis. And I don't know why. I don't know if it's because Newberg came second. I don't know if it's because Newberg was the only one publishing in English. I don't know if it was because Bauer lived in Hollywood, California, and no institutional affiliation when he wrote his. He was one of the leading geneticists, endocrinologists in Europe at one of the great universities in the University of Vienna. But he fled to the US. He worked at Penning at LSU for a year. I can't blame him for leaving Baton Rouge for Los Angeles when you come from Vienna. His son went on to become the dean at the USC Medical School, and he went on to work for the Hospital of Medical Evangelists, but he never had the influence again. Okay. This might not have happened, but by the late 1930s, we got the first animal model for obesity. Okay, up until then, we had the genetic model, Danforth's mice, but 1938-39, Albert Hetherington, who was a postdoctoral student working for Stephen Ranson, who was the leading neuroanatomist of the era, okay, at Northwestern University Medical School, develops a reproducible way to create obesity in mice using what's called a stereotaxic instant, okay? They do the experiment, we create the obesity, basically target a lesion in the ventral medial hypothalamus of the mouse. And by very careful positioning of this lesion, the animal becomes obese, often grossly obese after the surgery. Hetherington and Ranson write in 1940, invariably the fat animals have shown enormous accumulation of adipose tissue in the abdomen. As a matter of fact, the ease with which the results can be duplicated should make such rats a useful variety of standard test animal for physiological experimentation. These rats will also grow fat, though, in all likelihood more slowly, even when food intake is limited to an amount equal to or even a little smaller than that of normal litter meats. So normally when you create the lesion, the animals get obese, they wake up from the surgery starving, voracious. It's called hyperphagia. I'm going to discuss it then. But occasionally they don't, and they get obese anyway. And Hetherington and Ranson, particularly Ranson, found this fascinating. They said some animals with high food intake and some with decreased activity, the two factors are complementary in their effects upon body weight. Both would tend to increase it, but these two factors may only be symptomatic and not fundamental. By symptomatic, Ranson and Hetherington had read Bauer's article. They cite Bauer's article. And by symptomatic, I mean if the lesion creates a disorder of fat accumulation, fat storage, so that the fat mass is accumulating, equivalent to giving a young animal growth hormone. But now you're giving it something that makes the fat mass accumulate. The animal will have to That fat mass will take the energy it needs, and the animal will have to compensate by either being very hungry and eating a lot or expending less. And that's what they're suggesting. It is not difficult to imagine, for example, a condition of hidden cellular semi-starvation caused by a lack of easily utilizable energy-producing material, which would soon tend to force the body either to increase its general food intake or to cut down its energy expenditure or both. Now, if this line of thought had continued, it might have undone what Newberg did, but it did not. Okay, this is one of the jokes in this research. Stephen Ranson, as I said, was a leading neuroanatomist today. In fact, he had come from studying diabetes insipidus, which can also be caused by a lesion in the hypothalamus. And diabetes insipidus is characterized by excessive urination and excessive thirst. And the conventional thinking might be that the lesion causes the animals to be thirsty, they drink too much fluid, and they have to urinate all the time. But what Ranson learns is that the lesion actually creates a defect in an anti-diuretic hormone, vasopressin, that causes the animals to urinate all the time, the polyuria, and the loss of body water causes the thirst. The animal drinks to respond to the loss of body water. Here he's suggesting that the lesion causes some hormonal increase in fat storage. Energy is lost into the fat storage, hence internal hidden cellular semi-starvation, and the animal's hyperphagia is caused by that. 1943, Ransom dies of a heart attack. Albert Hetherington, in the midst of the war, joins the Air Force. And that's the end of this research. Except John Brobeck, who was a postdoc with Hetherington and Ransom in the late 30s, goes to Yale to study for medical school. While he's at medical school, he needs a job to pay his expenses. The laboratory is working in as a stereotaxic instrument. He starts doing ventromedial hypothalamic experiments. He creates obese mice, and he notices that they are hyperphagic, they are voracious. And his discovery is hypothalamic hyperphagia, the idea that if you lesion a ventromedial hypothalamus, you will create a ravenously hungry mouse, and the obesity is a result of the ravenously hungry mouse. Brobeck has not read Bauer, or at least he does not cite Bauer, but he has read Newberg. He cites Newberg as the evidence that these animals and humans get fat because they eat too much, and he has now found hyperphagia. So now we're going to move to the post-war. Hetherington and Ransom are dead. Brobeck is now the leading authority on VMH lesions. Expressed in physiological terms, is a problem in the regulation of energy exchange. The situation is still obscure. The question, why does the animal with hypothalamic lesions eat so much extra food? What determines how much we'll eat? What changes in the internal environment set the animal to eating, and what changes are associated with satiety? The first animal model of obesity has now become an animal model of overeating, of an eating disorder. And the question that Brobeck is asking, becomes the dominant questions ever since. So instead of asking what is it about these animals that make them accumulate too much fat, or what is it about humans with obesity which makes them accumulate too much fat, we ask, why do they eat too much? Okay, the leading theories of obesity in the 1950s and 60s are theories of food intake regulation. Okay, the glucostatic theory and the lipostatic theory. The glucostatic theory is promoted by Jean Mayer. It made a Harvard nutritionist. Mayer was a French immigrant who fought for the French resistance in World War II. And when I asked one of his colleagues, I never got the chance to talk to Mayer. When I asked one of his colleagues why Mayer never cited the German literature, although I was wrong, he actually did a few times. The colleague said, well, he didn't like the Germans. He killed quite a few of them. Mayer says it may be noted that the glucostatic theory seems to permit interpretation of certain types of alteration of the regulation of energy intake, in particular hypothalamic obesity, the hereditary obese hyperglycemic syndrome of mice, and at least one form of human obesity. The lipostatic theory is promoted by Gordon Kennedy at the British National Institute for Medical Research. Again, he's trying to explain the hypothalamic damage. The young rat adjusts its food intake so precisely to its energy needs that its rat stores remain almost constant. Hypothalamic damage permits excessive intake and causes obesity. It is suggested that the hypothalamic satiety mechanism is concerned only in the convention of an overall surplus of energy intake over expenditure, which would cause a deposition of fat. Okay, this is actually the basis for all our current thinking on obesity today and on appetite regulation, on neurocircuitry that controls obesity. I haven't looked at my watch, and I have no idea how late I'm running. If we have time, I'll get to it. 1965, the American Physiological Society actually publishes a handbook of adipose tissue, section 5. This is an attempt to take everything that has been learned about adipose tissue metabolism and put it in one volume so that the research community can make use of it. I don't say the obesity research community, but that is implied. Okay, it's 800 pages, it's 69 chapters, it's 4,100 references. Okay, it is dedicated to Ernst Wertheimer and Franz Hausberger, two founding fathers of adipose tissue physiology. There's only one reference to Newberg in terms of water retention. There's only 2 references to Gordon Kennedy's work. This is purely about the regulation of adipose tissue metabolism. And as they say, to put together to assemble current knowledge relating to adipose tissue, because it should be relevant to how we study obesity, and it has 0 effect. I think the problem is if you publish it in a volume, 900 pages, and physicians and researchers don't get that volume delivered on their doorstep, they are not going to read it, and even if they get it, they're not going to have time. 1973, the NIH hosts the very first obesity conference ever, the Fogarty International Center. Okay, it has 0 relationship with adipose tissue. Okay, now we are talking about obesity, we're talking about the control of food intake, we're talking about energy balance. They do talk about endocrine and metabolic adaptations to obesity, but the assumption is they are adaptations to obesity, not because of. There are no relevant causes related to what was in 8 years ago, this 800-page volume. Let me give you the leptin theory quickly, okay? Just so you understand how these poorly tested theories, once they become integrated into the literature and they're not criticized, how much damage they could do. In 1950, researchers at the Jackson Memorial Laboratory, Bar Harbor, Maine, discovered the OB mouse, the fat mouse. They published it in 1950. You can see in the photo, it's seriously obese. And then people start studying it. One of the fundamental things you might want to do with an obese mouse is find out not how much it eats, but whether or not it gets obese even when it doesn't eat a lot of food. Is it a fat storage disorder or an overeating disorder? So in 1953, Alonzo and Marin say long-term restriction of food in the genetically obese mouse decreases weight gain in growing mice. We restrict their food, they don't get as fat, decreases absolute weight in the adult. Although the restricted might have less fat than their unrestricted obese siblings, they still maintain body fat concentrations 2 to 4 times as great as their lean siblings who received 1.3 to 2.7 times as much food. Okay, these animals have a fat storage disorder. In 1961, the Zuckers, Lois and Theodore, published the rat equivalent of the obese mouse. Okay, the rat fatties and the hypoglycemic obese mice have a more basic metabolic disturbance than simply an increased appetite. When restricted to a normal food intake, they are still obviously fat. Therefore, in these 2 cases, it indicated that there was an abnormality in intermediate fat metabolism. Okay, then Douglas Coleman gets involved. Douglas Coleman did the parabiosis experiments, which Jeffrey Friedman then based his work on to clone and identify the leptin gene, which then became the basis. How we think about leptin became the basis of how we think about the neurocircuitry of appetite and the cause of obesity. Okay, Coleman, in a paper that's been published over 1,000 times, discuss what happens when you take these OB and then DB mice, which have a defective leptin receptor, and food deprive them. The percentage of body fat accumulated in both obese mutants was increased to values similar to mutants fed ad lib. Even under these conditions of relatively food deprivation, decreased energy expenditure of all these mutants, although undoubtedly a factor, cannot totally account for this increased food efficiency. It's not just that they're expending less. It must be concluded that although hyperphagia is always present under food ad libitum conditions, it is not a necessary prerequisite for the development of obesity, which is exactly what Malcolm Fleming said about humans in 1760. In 1985, Coleman publishes an article in which he literally pair feeds the obese animals with lean mammals. He feeds them at the same time as the lean animals, and he gives the obese animals only half the food that the lean animals need, therefore guaranteeing that they are starving. And even when maintained on 50% of normal food intake, mutants still become obese. Okay, these animals have a fat storage disorder. What you want to know is why do they become fat even when they are semi-starved? They're also grossly hyperinsulinemic, which might be a sign. I'm not going to get into this. Jeff Friedman, heroic molecular biologist at Rockefeller University, begins the attempt to clone the gene responsible for the OB mouse in the mid-ish 1980s. It takes him a decade. The reason for doing it is because he is basing his thinking on Gordon Kennedy's lipostatic idea. He is looking for the molecule that signals from the fat cell to the brain how much to eat, tells the brain to stop eating if you believe that hyperphagia is the cause of obesity. Friedman's thinking is basically Newberg, Brobeck, Kennedy interpretation. And he starts the abstract of the paper that announces the discovery of leptin, the mechanisms that balance food intake and energy expenditure to determine who will be obese and who will be lean. The energy balance in mammals was therefore postulated to be controlled by a feedback loop. He's now talking about Kennedy's hypothesis, in which the amount of stored energy is sensed by the hypothalamus, which adjusts food intake and energy expenditure to maintain a constant body weight. Our results, particularly the evidence that the OB protein product is secreted, suggest that OB may encode this circulating factor, consistent with the lipostasis theory, implying that an increase in the level of OB signal may act directly or indirectly in the CNS to inhibit food intake and or regulate energy expenditure. What it cannot do is explain why these animals would get obese even when half starved. Now you've controlled for satiety, you've controlled for hunger, you've guaranteed that the animals are hungry because you're semi-starving. The New York Times, when they published the leptin discovery on the front page, middle column, and that way the fat stores in the body tell the brain how big or small they are, and the brain can regulate food intake accordingly. Last year, Friedman writes an article on the causes of obesity and its treatment. The end of the beginning, he's connecting the leptin thinking to the GLP-1 thinking, and it is the same thinking. The discovery of the causes of obesity and means for treating it also revealed that obesity is a brain disorder. Each of the single gene mutations that are known to cause obesity in humans alters the function of repetitive pathways in the brain, whether you're hungry, how quickly you get satiated, as do the new treatments. This has led to an explosion of information about the neural circuits that regulate appetite, and feeding is arguably the best delineator of complex behavior. What is absent there is any discussion of what leptin does in the periphery to actually increase fat storage, which is a fundamental observation. And we know that fat storage increases even when the animals are half starved. When I asked Jeff Friedman about this and Coleman's observation, he said this was 2022. It's an interesting point. I haven't given it a lot of thought. The time for one question. Do I have time? The impact on the community? Let me just give you the two paradigms, okay? Because they're fundamentally different paradigms, as much as thinking that the Earth rotates around the sun or the sun rotates around the Earth. On the left is the energy and energy outdrives fat storage. On the right is fat storage, dysregulation of fat storage drives intake. So obesity is an energy balance disorder versus a fat storage disorder. How much you eat and exercise is critical. What your body does with what you eat is critical. That's intermediary metabolism. Does your body prioritize fat storage, or does it prioritize energy expenditure? Because if the latter, then you're going to be one of those people who's out riding his bike 2 hours after lunch, If the former, you're going to want these people taking a nap after lunch while you get fatter. Calories are critical. How much? Macronutrients, okay? High GI carbs and sugars. What you're interested in is how the foods you eat influence the hormonal regulation of fat storage and metabolism. They do so very differently. Fats, proteins, carbohydrates, and sugars. And that's what you study. Neurocircuitry per Jeff Friedman is appetitive behavior and expenditure. And here the neurocircuitry works through peripheral metabolism and storage, and that in turn influences behavior. Energy balance, the brain is supposedly in control of the body. And in this alternative hypothesis, the body basically is driving the brain's behavior. And if you want to prevent obesity, you are fighting your body's urge to do so. You are not in control of it. Anyway, thank you very much. I appreciate your patience and your time. Acknowledgements and further reading. These two papers on the left were written by myself. The paper, Trapped Fat, Obesity Pathogenesis and Intrinsic Disorder Metabolic Fuel Partitioning, was published 2 years ago. It's the science, it's all the animal experiments, all the major animal models, and what they do or do not show about the sort of energy balance versus trapped fat hypothesis. The paper in the middle, the carbohydrate insulin model, is the dietary implications of this theory, which is that the problem with modern diets and with getting fat is in the carbohydrates you're eating, not the amount of food. And then the paper on the right was an attempt, it was a symposium hosted by the Nobel Nordis Foundation to bring together the people who believe that obesity is a fuel partitioning fat storage disorder with the people who vehemently don't, and to come to some kind of discussion about what needs to be done. I'm grateful for every one of those papers, even the ones that vehemently don't, because they care enough about this very important subject to argue with us. Not everyone does. Thank you.
We have time for maybe 5 minutes of questions, and I promise I will. Thank you for that talk. Why don't we open it up for questions in the audience, and then I'll finish with the audience, the last question.
Gary, so before writing on nutrition, you did a lot of work on code fusion. How do you think the culture in physics, for example, seems to differ from the culture in nutrition research? Did you notice any differences?
Yes, thank you. I cut my teeth in as a journalist studying bad science, pathological science. I mentioned, which is technically, it's a term coined by the Nobel laureate Irving Langmuir for the science of things that aren't so. Okay, scientists screw up all the time. What I'm describing is a screw up that happens to have lasted in the literature with tragic circumstances for 60 years. The way to prevent that from happening is to vigorously criticize and basically attack every paper, every idea. The researchers are supposed to do it themselves. I often end up quoting Richard Feynman in my writing. The first principle of science is that you must not fool yourself. You're the easiest person to fool. So the idea is in a functioning science, what you're thinking about is how you screwed up. What you stay up at night worrying about is how you screwed up, and you value the people who disagree with you, who think you screwed up, because they're going to tell you why and how. And then we allow you to do better experiments to find out whether you're fooling yourself or not. When I first started writing about public health science in the early 1990s, I started with I did electromagnetic fields from power lines cause cancer, and I was writing about epidemiology. And I had come from the physics world. What struck me the most was the absolute lack of critical discussion about not just the science itself, the evidence, but the methodology. Methodology was being used to try and establish cause and effect that it didn't appear to be able to do, and nobody was arguing. Nobody, if in the physics world where I grew up, and again, the physics world in which I grew up was a world that was dominated by European emigres still. or the students of these European immigrants. They had a way of thinking about science. When you go back and read that, as I've been doing, and I have to think the kind of research I do is only possible because of AI, because I can translate all the German literature without going bankrupt. And I can do it almost instantaneously, at least Claude can. You read this literature, it's brutal in its critique of everybody's work. And they don't mention papers. It's not referenced papers as though they're somehow factoids that are out there without promoters of these ideas. They discuss the people who did the research. They discuss why they don't believe the research. They discuss what other research shows and the problem with that methodology. If you don't have that kind of critical, hypercritical world in science, I think it ceases to function. And then when you get so far along with ideas or methodology that you have entire disciplines built on them, there's nothing to be gained by critiquing them. Whoever does so is just going to basically talk themselves out of the field. So you have a kind of fundamental systematic problem, and the end result is people like me getting involved in the current, your bosses, who think pathological science are everywhere because they know that it's certainly a problem in some places.
Hello, I think that's a great talk. I really enjoyed it. But in this era of personalized medicine, isn't it possible that both can be true? So, for example, there's me and my husband. I can eat anything, and I will never gain weight. My husband can eat like a bird. He has to bicycle 50 miles a day for a month to lose 5 pounds. So I think there are genes and certainly different metabolisms. And certainly I agree, different fat depots as well. Like my family is all apples. His is not. He's still having these issues. So I think that, people look at what they look at and they draw their conclusions. Somebody else looks at what they look at, they have their conclusions, and maybe they're both right in a way.
This is, one of the problems with this is the idea that fat storage is biologically determined seems obvious. Okay, all you have to do is look around when these physicians in the 19th century are making this point that the world is full of people who get obese who do not eat a lot. They didn't need an epidemiological survey and food frequency questionnaires. They just had to look at their friends and their relatives, their colleagues, their co-workers. The world is full of people who try to restrain from eating, who eat less than I do, and then grow obese. So you have to explain that. And yet we have a theory of obesity that cannot and makes no attempt to. Okay, because people who got too fat at some point or another, they took in more energy than they expended, which is obvious because they gained weight. It's true of everyone who gets larger, taller, fatter, more muscular. And we've let this theory stay out there. And one of the things that I found interesting in doing my research was, so the German thinking vanishes with the Second World War. It remains through the 1960s in this sort of science of fat metabolism, but even that is slowly evaporating. And then in 1967, the concept of health at any size is created. A freelance writer writes an article for the new Life magazine, basically saying, look, both me and my wife are fat and we are tired of trying to not be. We are tired of the insinuations that we should be thinner, that we should be starving ourselves all the time. And so the critique, the pushback does not come from the scientific community. It comes from people who live with obesity because they know what the truth is, but even they have to try and think about it in terms of, do I just eat too much? Am I too hungry? Is it my problem? When the reality is, it is, you are living with a body. What's her name? The New York Times columnist, J, that talks about her unruly body. The body that wants to be obese. So the question is, how do we deal with that? We have to study to understand that. It's not how much people eat and exercise.
Thank you very much for a very thought-provoking lecture, both the direct topic and the implications for many other areas of science. I guess I had a question for you about your thoughts about how do you take that sort of fundamental argument of these two potential paradigms with the delta E on two different sides of the equation, and understand the tremendous increases in the last 40 years, particularly in the US.
And that's right, a fundamental question. That's what I was trying to understand when I got into this.
What's your answer now?
So again, when I've spent 25, went from being a journalist to de facto medical historian for, I don't know if I do it well or not, but that's what I do now. Back in the 1950s and 1960s, the British nutritionists, British physicians around the world were trying to understand the appearance of obesity and diabetes in the populations that they administered to. They had the British Empire with its colonial hospitals, missionary hospitals all around the world. All around the world, basically, they were seeing the same phenomena. Whenever a population became westernized, when it came into contact with the West and traded with the West, you eventually saw an increase in obesity and diabetes. And they had the Western population, whatever it might be, loggers, missionaries, to compare rates with. And the conclusion was that when Western populations come into contact with the isolated traditional populations eating whatever they're eating, they are trading sugar and white flour. And so you ended up basically by the 1960s with competing hypotheses